Thank you, Louie. And thanks to the organizers for giving me an opportunity to participate and present and talk about what we're doing in Cleveland. I guess as the last speaker, my job is to give you one final dose of inspiration in what has been really a very inspirational meeting and event. And I hope I can do that. I was asked to talk a little bit about the environment in Cleveland that's supporting cancer therapeutics development. So I'll tell you a little bit about our space, the resources we have, and then to give a few examples of the types of therapeutics development efforts that's supporting. Not all about my program. I'll highlight a few some work that's being done by some other investigators. As Louis mentioned, I'm the division chief in PT Monk, but I am the director of an institute that's named the Angie Fowler Adolescent and Young Adult Cancer Institute, and it's named after this young girl, Angie Fowler, who wasn't actually treated at Rainbow. She was diagnosed 41 years ago with a very rare and aggressive melanoma. It was 1983. I was a medical student then. The options for therapy for melanoma at the time were nothing like they are today: surgery, interferon alpha, and it was metastatic at diagnosis. She died 3 months after diagnosis. And her parents, Chuck and Char Fowler, were very determined that teenagers going through that experience would have a different environment in which they could receive their care and potentially better options for therapy. And so many, many years later, living in Cleveland, they personally have given over $30 million of their own investment to support environment and research around adolescent and young adult cancer. But more important than that, they've done exactly what you've seen many other people here do this week, and that is they were really willing to tell their story over and over again about their experience. And that's inspired many other people to get behind the mission and our program at Rainbow. So I hope I can convince all of you to come to Cleveland and to see this incredible space. I want to acknowledge Phil and Wendy from Little Princess Trust who are here. They recently made the trip to Cleveland, and I hope they'll share with you their experience that they had. So again, I'm going to just do 3 things in my talk. One is to tell you a little bit about the environment that's supporting cancer therapeutics development in Cleveland, what that looks like. I want to give a few highlights of the drug development effort that's supporting, not all about my program, but what some of my colleagues are doing in the cancer space. And then to just highlight again the value of partnerships. You've heard over and over again about the importance of academic industry partnerships. That's truly important. I want to emphasize the importance in our space of the partnership between pediatric and adult cancer programs. That helps us to move more rapidly. It also helps us to bring greater infrastructure supporting what we're doing. And I'll tell you what that looks like in Cleveland. And again, the importance of our patient partnerships. These are my disclosures. As everything else in life, this is about relationships. The gentleman on the top, Sunjin Kim, was a member of the research lab I worked in for many years, the National Cancer Institute. Our labs were right across the hall from each other for about 15 years, where we were studying and focused on the biology of a cytokine known as transforming growth factor beta. Plays a very important role in tumor immunology. And we were both mentored by this gentleman, Michael B. Sporn, who founded the Laboratory of Chemoprevention. Listening to Sarah talk, I had no idea what your topic was going to be. In thinking about his mission and his focus, I think he would have enjoyed your presentation today. Sungjin left the NIH around the same time I left for Cleveland in 2006. He went to start a new institute in Incheon, but eventually crossed the road to entrepreneurship and started MedPacto, and they're leading the development of a first-in-class inhibitor of the type 1 receptor for TGF-beta. Mike crossed the road to entrepreneurship a little bit later in life at age 87, my hero, and started this company called Triterpenoid Therapeutics, and I'll talk a little bit about those agents that are under development. So this is Cleveland. As Louis mentioned, I'm at Rainbow Babies and Children's Hospital. That was founded over a century ago by 9 young women who were from prominent and affluent families in Cleveland, and their mission was to develop a perfect determination to relieve suffering. That children's hospital over that century and now 125 years has been an important safety net for pediatric patients and families living in the Cleveland community. But beyond that, it's been home to many academics, physician scientists who've really changed the standard of care for patients in neonatology, cardiology, many other fields. So it's a wonderful home for what we do. And in the pediatric cancer program, we benefit from our partnership with what's known now as the Seidman Cancer Center, named after donors who helped to build, through a huge gift, this freestanding cancer hospital entirely dedicated to the care of adult patients with cancer. But the resources that those two entities bring together to support cancer care and cancer research is really phenomenal. Examples of that include a proton therapy center that's right in the building, so our pediatric patients don't have to go a long distance to access it. And I'll talk a little bit about a GMP cell manufacturing facility, 10 ICOS, 7 clean rooms that are supporting GMP manufacturing of cell products. And then important to what I mentioned with respect to the Fowlers, we're in an era now where pediatric cancer is defined by a molecular fingerprint, not your age. So there are many patients who are in their 30s, 40s, and even 50s who have pediatric cancer. So the expertise that exists within Rainbow allows us to offer those patients an opportunity to participate in clinical trials in a facility that's age appropriate for them. The other thing I just want to touch on, you've heard a lot about the journey of patients with life-threatening illnesses and the chronic nature of those illnesses. They spend a lot of time in the hospital. And one of the things that was really important to us and to the Fowler family and many others is that the environment in which we provide care really matters. We've talked a lot this week about what we do, but how we do it and where we do it matters a lot to patients. Their gifts have really helped us to transform the top 3 floors of our children's hospital into really a magical space. It's a wonderful space that's capped by this rooftop garden and sky lobby, which is just a wonderful space for families. And to show you just how close our connection is with the UK, when we had our ribbon cutting ceremony for this, we actually had Roger Daltrey of The Who come. Many of you know that here, his work with Teen Cancer Trust helped to build some really wonderful facilities across the UK, really dedicated to offering a better experience for teens and young adults with cancer. When the CEO of Teen Cancer Trust came to the US and started Teen Cancer America, Roger got very involved. This is Chuck and Char Fowler. They became members of the board of Teen Cancer America, and that built a very wonderful relationship. He came to Cleveland, actually played a benefit concert for this facility, and has been there to visit us actually multiple times. And then this is the Seidman Cancer Center. It opened its doors in 2011. It's really a remarkable facility. It's about 375,000 square feet, 120 beds. It's the home of clinical care for a major part of what goes on in our NCI-designated comprehensive cancer center, the Case Comprehensive Cancer Center. Thousands of accruals to clinical trials, much of which happens in this space. And again, a really valuable thing for us to be a part of it. And now in the immunotherapy world, that's supported by the build-out of what's known now as the Wesley Center for Immunotherapy because of a $10 million gift from the Wesley family just a few years ago. But it didn't start in 2020. Actually, the inception of the cell therapy started way back in the 1990s because of the discovery of mesenchymal stem cells at Case Western Reserve University in Cleveland by Arnie Kaplan, who passed away this past year. But that discovery of MSCs and the realization that they could be used to treat many diseases like IBD and to treat graft-versus-host disease inspired the construction of this facility. There was investment from the federal government, from the state of Ohio, to create what was called the National Center for Stem Cell and Regenerative Medicine. That facility had 6 clean rooms. It's now, because of the gift from the Wesley family, a 10 clean room facility. Also has capacity for benchtop and closed system manufacturing of CAR T-cell products. So just a wonderful space, and it's associated with about 3,600 square feet of research space that has yet to be filled out with investigators. So come to Cleveland and see it. Okay, so I'm going to give you just 4 short vignettes, examples of the type of therapeutics development effort that has happened because of that collaborative environment within Cleveland. And actually, the first one I'm going to tell you about isn't new. It's actually an old story. All of my colleagues in pediatric oncology know about the use of anti-GD2 immunotherapy for neuroblastoma and about the development of Unituxin denitumumab for that use. But what you might not remember or know was that the very first phase 1 clinical trial with that was actually in Cleveland. At the time, it was a very young physician scientist, Naikon Chung, who's now at Sloan Kettering, who was developing a mouse monoclonal the F3F8 antibody, and wanted to do a phase 1 safety trial in pediatric patients with neuroblastoma. That would have taken forever if they didn't include adult patients. And so he partnered with this gentleman, Nate Berger, who at the time was leading the division of hematology oncology. And so they partnered to do this trial that included about 17 patients with both neuroblastoma and melanoma. Multiple patients had a complete response to the antibody and several partial responses, which inspired further development of that agent. Now, a couple things to note on this slide. The first is if you look at the year this was published and the year of the FDA approval of that antibody for pediatric cancer, it was 30 years, right? So fortunately, I think we're doing a lot better than that today, but our patients deserve better, right? We need to be much quicker in developing these therapies. The other thing I want to point out again is the importance of this collaboration between a pediatrician and a physician who focuses on the care of adult patients. Nate was an extraordinary person. He was the director, the inaugural director of our NCI-designated cancer center. He actually founded and established the first HEMONC training program for physicians in Cleveland. I actually called him when this was FDA approved to thank him for helping this pediatric oncologist to do that first phase 1 trial. And, you know, as an incredibly accomplished person, to show how humble he was, he said, "It's nice to know that somewhere along my career, I actually did something worthwhile," which I thought was really amazing. Unfortunately, we lost Dr. Berger this year. He was a giant in our field. The second example is current. So flash forward now 30 years, and these are two of my colleagues in pediatric oncology. Alex Wong, I first met when he was a pediatric oncology fellow at the NIH. He was in a combined Johns Hopkins NCI fellowship training program. He trained in the laboratory of Drew Pardoll, who many of you know is a giant in the field of immunotherapy. Alex is a consummate tumor immunologist. That's his expertise. He actually, in our cancer center, is the director of the Immunology program in our comprehensive cancer center. He also directs or leads what is known as our MSTP or MD-PhD training program. He's a wonderful mentor. And one of his mentees is Kristen Van Heist. She's a clinical investigator who's responsible for developing clinical trials within our pediatric oncology program. Alex is very focused on osteosarcoma as a disease. Most of his research is focused on that cancer. Years ago, when we were at the NIH, we were looking at the expression of transforming growth factor beta in childhood cancers. Osteosarcoma, as a mesenchymal tumor, is one where the expression of that cytokine is very high. And our work showed in the 90s that it was a mitogen, actually an autocrine growth factor for those tumors. But Alex's idea was that its production by those tumors actually was one of the key factors responsible for creating this immune desert within the tumor, and the reason why immunotherapy might not work well. So he wanted to prove that case, and he said, you know, is there any way we can strategize and use an inhibitor of the TGF-beta pathway to study that? So we connected with MedPacto and Dr. Kim and obtained Vactocertib and a little support from them to test that in some preclinical models. And again, this is a small molecule inhibitor of the type 1 receptor, which is the signaling heterodimeric complex that is responsible for transcribing the signal from the receptor. And so to do that, he used two well-established preclinical models of osteosarcoma, one that involves use of human osteosarcoma cell lines or PDX models grown in a immunocompromised or NSG mouse models where you can't really assess the contribution of the immune system to response. And the other is a syngeneic mouse model where you have a competent immune system, so it's a mouse osteosarcoma in a mouse. In either case, exposure to Vactocertib really suppressed the progression of osteosarcoma. So here on the left, you're looking at an osteosarcoma that was established in mice, and after 67 days, they were started on a daily dose of Vactocertib, and you can see that it really suppressed the growth of those tumors and knocked out the activation of the signaling intermediate SMAD2. And in the syngeneic mouse model, one of the hallmarks of disease is metastasis to the lungs, which is really driven by inflammation within the lungs or the accessory immune cells that suppress immune response. So it really wiped out the metastatic spread, and that was associated with an increase in infiltrating interferon gamma producing CD8 positive T cells and a reduction in CD4 T cells that express PD-1, again, pointing to the possibility that inhibiting TGF-beta may overcome T cell exhaustion in those models. So that was enough to encourage the company to enable a compassionate use exemption, a single patient IND, and to capture an orphan disease designation from the FDA. This is the very first patient, Izzy, who went on that drug as a compassionate use. Izzy had a very high risk, aggressive osteosarcoma, progressed right through his upfront therapy of a lower limb osteo. He actually had to have an amputation to control the primary tumor. And then, as many patients do, he suffered repeated relapses. The initial ones were within the lung. And the natural history for any of these patients who have a lung metastasis is they live for about 6 months with repeated metastases. And that was Izzy's story. The relapses were 1 to 2 months apart. He went on Vactasertib as a compassionate use and then went for a year and a half without another recurrence of his disease. Eventually, it grew through that, and he's passed away. There were an additional 2 patients who started on this through compassionate use, but last year, we were granted an IND, or Alex was, and Kristen were granted an IND to establish the first clinical trial of this. It's a multi-center trial. It's multiple institutions, and patients over the age of 14 with osteosarcoma will be able to participate. All right, the second story is similar to the work that John's doing, and this is developing a cell-based therapy, a CAR T-cell therapy. The individual responsible for developing this technology is Reshmi Parameswaran. She is a PhD at Case. I first met her when she was just beginning her career, and she was interested in targeting TGF-beta as she in the use of NK cell therapies and CAR T-cell therapies. So we talked a lot about that. She was able to get a Career Development Award, a 5-year award from the St. Baldrick's Foundation that was helping her to develop these cell-based therapies. And one of the technologies that she created was a CAR T-cell expressing BAFF ligand, which is important for the viability of B cells. And it does so by acting with multiple receptors on B cells. So it's a viability factor. And her idea was that if we could express BAFF ligand on the CAR T, you could overcome the possibility that you might lose expression of one antigen or another, right? So you could overcome therapy resistance. So she's tested that in a mouse model. The data were convincing enough to encourage investment in partnership with Luminary Therapeutics and Novartis. And that has now gone into clinical trial. I'll highlight that in the next slide. I just also wanted to point out that one of the things that HDI has done, which I think is really valuable, is to support trainees. They have a program supporting MD, PhD students, and one of their scholars was working in Dr. Parameswaran's lab, learning the development of CAR T technologies, and I hope to see the HDI do more of that, including here. So this is what the construct looks like, and this is the trial. The very first development of this, again, happened in partnership with Luminary, and the plot on the right, on the top here, C, shows you the effects of this CAR T in human large B-cell lymphoma in a mouse model. That went on to encourage the development of this in a clinical trial. This is actually one of the first patients to receive the CAR T, patient with a diffuse large B-cell lymphoma. You can see it here on the PET scan, and 30 days later, that's gone. So it's a very valuable asset in our armamentarium for patients with B-cell malignancies. So the impact here is, again, another partnership with industry to create a novel CAR T therapy. Again, that's in 2 trials now. One is non-Hodgkin's lymphoma. The second is multiple myeloma. Those are both in Cleveland. But there's already a trial that's waiting to open to use this to treat patients with autoimmune disease like lupus. And we hope, of course, to be able to bring this into the pediatric age group as well for patients with B-cell malignancies. And then the last vignette I'm going to give you is focused on a relationship that I have with my former mentor at the NCI, who, as I mentioned, was very important in developing strategies for cancer chemoprevention. So if you look in the Oxford English Dictionary, Mike is credited with coining the term cancer chemoprevention and also coining the term retinoids. His whole focus was on the development of orally bioavailable agents that are safe for chronic administration, non-toxic, for the sole purpose of reducing the risk for cancer. And you think about what tamoxifen and other drugs like that have done for women who survive estrogen receptor positive breast cancer, the idea was that you could expand that and make that more valuable for the one reason that we heard about is that cancer is a process that develops over many, many years. That was Mike's passion. When I was a young fellow in the pediatric branch and I walked into his office and said, I want to join the lab of chemoprevention, his response to me was, you can't prevent cancer in children. And I said, let me tell you a story. And I told him the story of one of my patients who had leukemia. His first cancer was osteosarcoma when he was in his teenage years. He was cured of that, went on to develop a melanoma. He had surgery, interferon alpha, cured of that. Then he developed a therapy-related leukemia, went through bone marrow transplant, only to suffer another relapse and eventually die from his disease. The problem is we tell patients like that, you have cancer, we're going to cure it. You're at high risk for it coming back, but we do nothing in between to reduce the risk for recurrence. And our problem is everything we do to cure that first cancer puts them at higher risk for another cancer, and our opportunity to cure it with each successive cancer is less, right? So I told that story, and he said, okay, you can come in the lab. I did a lot of work studying TGF-beta, but started on a journey with him developing a new class of agents known as synthetic oleanine triterpenoids. These are synthetic derivatives of a natural product called oleanolic acid, which has many anti-inflammatory properties. Mike's idea was that inflammation is a driver of progression, right? So initiating events then have to be supported by progression factors, and inflammation in cancer is a big driver of that. So this started in the early 90s. The whole medicinal chemistry campaign was to optimize the activity of oleanolic acid, and applications were focused on neurodegenerative disease and cancer. This picture was actually taken in 2019. Mike and I were here in Liverpool visiting a collaborator and then making a trip to Almac, UK, because they were about to embark on a 2-kilogram synthesis of the lead triterpenoid. And he was doing what he loves to do, draw these new structures. I wish I would have kept this napkin. It was like artwork, but he was always coming up with new ways of developing and derivatizing these triterpenoids. He's really quite something. And this is really how this whole effort began. So the idea was to continually, through an iterative process, modify the structure, and then to evaluate how that would suppress key markers of inflammation. The principal assay that we would use would be the suppression of inducible nitric oxide synthase in primary macrophages. And you can see that second or third generation triterpenoid was very potent in doing that at very low nanomolar concentrations. And you can see on this plot how each successive change in the structure led to an increase in the ability to suppress inox expression to the point where we were over 10,000-fold more potent than the parent molecule oleanolic acid. CDDO-ME became known as bardoxolone methyl, and the top 3 compounds along the top here were all licensed in 2004 to a biotech company in Dallas called Reata Pharma. Reata was in the cancer space when I first started in Cleveland. We were actually doing clinical trials with CDDO and bardoxolone in cancer patients, but as a small biotech company, they knew the competition was tight in the cancer space, and they realized many patients on these drugs had improved renal function. So they left cancer and then went all the way through phase 3 trials in chronic kidney disease and Alport syndrome with bardoxolone, only to be denied an NDA at the end of that. But they persisted and didn't fall, and actually developed this fluorinated derivative, which was known as amavoxolone, but last year was approved by the FDA for Friedreich's ataxia. An incredible story. We worked with other fluorinated derivatives, which they've developed. They're very potent anti-inflammatory agents. We've shown efficacy, for example, in models of multiple sclerosis. But Mike wanted these to be developed for cancer, and he knew that the imidazolide was the most potent of all the triterpenoids that he had developed. And so at age 87 in 2018, he formed a company and came to Cleveland. He would come literally about once a year, and we'd meet with the entire lab and collaborators and talk about what we were doing with these compounds. And he walked into my office proudly proclaiming that he started a company. What are we going to do? And my answer was, we're going to need to find some money. So we embarked on writing a number of small business grants to fund the work that we're doing. Just a couple of points about this class of compounds. It's very unlike your typical lock and key, classic drug, one target. These molecules have the capability of interacting with a number of proteins. The work that's been done over the years has demonstrated that they react with thiol groups on a protein called KEAP1, which is an ubiquitin ligase. And KEAP1 targets a transcription factor known as NRF2 for degradation. But the ability of the triterpenoids to bind to KEAP1, because it's like a redox sensor, activates a whole class of antioxidant and stress response genes that are regulated by NRF2. But the anti-inflammatory properties are linked to its ability to bind to IKK beta and to block NF-kappa B signaling. So these are agents that, Mike used to like to describe them as network agents. They weren't necessarily target specific, but the selectivity is based on the carbocyclic skeleton that defines which proteins they interact with. As pediatric oncologists, we use multiple drugs in combination with different mechanisms of action to treat cancer. I don't think it's a negative to have one drug that has distinct mechanisms of action that could be used in the context of prevention, but it does present a challenge when you're talking about drug development. Because we knew these were very potent at regulating macrophage biology, our idea was that we could use these to interfere with the suppressive effects of macrophages in cancer. Macrophages are well known now to drive therapy resistance in the context of radiation therapy, conventional chemotherapy, and now even immunotherapy. And 2-piimidazolide is 1,000-fold more potent than the methyl ester or bardoxolone methyl, both in the suppression of iNOS and of CCL2. And the view was that the ability of the triterpenoid 2-PM to suppress this conversion from M1 to M2 could be beneficial in the context of cancer metastasis and progression. So we chose to focus on high-grade glioma because of the understood relationship between the infiltration of macrophages within the tumor and high-grade disease and therapy resistance. This has been demonstrated over and over again looking at markers of M2 macrophages. This is an analysis of RNA-seq data from the TCGA database, and this is immunostaining for CD206, which is an M2 macrophage marker. But to start out, we actually looked at activity in preclinical models, and this includes in vitro models looking at whether or not there's single-agent activity of this triterpenoid in high-grade glioma. What I'm showing you here is the response of 2 glioma stem cell lines, including C475, which actually is a multi-drug and radiotherapy-resistant glioma stem cell line. And you can see that even at nanomolar concentrations, this drug is very active in suppressing the viability of these glioma stem cells. And importantly, exposure to 2-PM actually enhanced the response to radiation, so the potential that it could act as a radiosensitizer became something of great interest. So we went on to look at this in other high-grade gliomas, including the rare pediatric glioma known as DIPG. This is diffuse intrinsic pontine glioma. And using a colony-forming assay where you plate these cells at low density, wait a week to 2 weeks, and then look at the number of colonies and the size of colonies, we can see, again, very low concentration of 2P actually suppresses colony formation. And when you combine it with radiation, 2 gray of radiation, it does so even more. So active as a single agent. We've gone now to actually evaluate that in vivo. So again, DIPG is a rare tumor. Typical age of diagnosis is between 4 and 11 years of age. There are less than 400 cases in the entire US every year, and it's uniformly lethal. You can't do surgery because of where it's located. We use radiation therapy, but it's largely palliative, and most patients don't live more than 9 months. So the fact that we can see activity in vivo is very encouraging. Big next steps are, can we combine that with radiation? Can we get durable long-term responses to the drug? To summarize this last part of the talk, we know that CDDO2-PM is a synthetic triterpenoid that effectively crosses the blood-brain barrier. That's now been duplicated in other preclinical models, not of cancer, but neurodegenerative disease. Our collaborator, Yamamoto in Japan, has actually used this. Yamamoto is a leader in the field of NRF2 biology, has used this in 2 different preclinical models of Alzheimer's disease. He's again shown its ability to cross the blood-brain barrier and to prevent disease manifestations in these preclinical models of Alzheimer's. This was just published this year. We have activity in high-grade glioma, including DIPG. What I haven't shown you is that it's not toxic to normal astrocytes, and we've shown that using primary cultures of brain astrocytes. And again, it's orally bioavailable. So to conclude, I just want to, again, highlight the importance of collaboration. And I also just want to say to the young people in the audience, the importance of a good mentor. And if you're lucky, somewhere along your career, you'll find a mentor who will be a lifelong colleague, collaborator, and friend. Mike certainly was that for me. This week is actually the second year of his anniversary of his passing. He was a giant. If you want to read about him, there was a great perspective written on Mike in Nature Index in 2017. And then I just wanted to share this email that was from Anita Roberts. Anita was one of his mentees who came into the NIH at a very young age as a postdoc, became a partner for Mike Sporn. When I left or was leaving the NIH to go to Cleveland, she was suffering from her own battle with gastric cancer. And when she was cleaning up her office, she had pulled down this email that was pinned to the wall that she saved. It was an exchange between her and Mike. On his return from one of his many trips in the early 80s, meetings where we were talking about TGF-beta, he sent this email, always thinking about what's next, and he said, Anita, our next dimensions, to be continuously creative, to be productive as possible, to be innovative, to see new perspectives, to see things as no one has seen them before, to have our creations last, and then most importantly, to relieve human pain and suffering. And that's why we're all here. And I just wanted to share that. I thought that would be a nice way to end the meeting. So thank you all for letting me present.